finishing up with sensation and perception, focusing a little bit more in detail about perception. And we have our expert in the department, Heather Mong, here to talk with you about perception today. So give her your undivided attention. Will the stuff that she talks about be on your exam? Yes. All right.
Great.
Here you go.
Hello, guys. Good afternoon. Okay. I am Heather Mong. I am also a fifth year in the cognitive program. I study visual attention and learning, so I love sensation and perception. I think vision is awesome. Vision is good for everybody. Okay, so also I'll note that I am wearing a wire. We're videotaping this, so do ask questions. Don't be shy. But that's what the is. Also, do ask questions. Stop me. I will just fly right by something. If you don't get it, let me know. If I don't see you, just raise your hand like that.
Can you speak up a little bit?
Yeah. Okay. So if you don't get something, stop me. Raise your hand. If I don't see your hand go up right away, start waving, pointing, whatever. I tend to be fairly good, but I can't guarantee I'll see you. Okay. So what we're talking about today, I'll start with a little bit of jargon. This is important because I'll just go into talking about cells firing like there's no tomorrow. And it's important you understand what I'm talking about there. We'll do a review of what you covered Monday, and especially for where it's relevant for today. We will talk a little bit about naive realism, both what it actually is and how it's relevant for perception. Then we'll go through a couple errors in your perception. It worked in the last class, so hopefully we'll be golden. And then some fun surprise stuff at the end. Okay. So, jargon. The jargon I use in this lecture is basically cells firing. This is basically what you talked about with action potentials in neurons. Cells communicating to each other. One cell sees something happen, trying to communicate that to the next cell. It's firing. This is specifically for cells responding to an event that they like. So an example from Monday is photoreceptors. They like light. Light hits them, they fire. So that's the sort of thing we're talking about. Okay, so to make sure we are 100% on the same page. Yeah. Okay. So hopefully this is a review from your neuropsychology section. I'm not really concerned about how the cells fire, the whole ion gating, response curve, that stuff. Not really relevant for this. Just the fact that this is how cells communicate their information. Okay, hopefully that was enough time on that slide. Okay, we're going to pretend that this little pirate guy is a cell. He's a happy cell. Okay, so something happens that this cell likes, like light hitting it. The cell fires its little cannon to communicate to its other little pirate cell friends something happened. Cell needs time to reload, so it can fire again. It can fire again. This can happen frequently. But this is basically what we're talking about when a cell fires. It sends information along to its little cell friends. Good? Okay. So a couple other important things are the base rate of firing. So a cell doesn't just sit there not doing anything if it's not being used. It will fire just periodically, kind of accidentally. And we call this sort of accidental rate of firing the base rate. This is it just going off on its own, sort of like accidentally shooting off a cannon. Also, cells have an all or nothing response. That is, they don't send off half a cannonball. They can only send off a full one. And so what this means for how strong a cell is firing doesn't mean it's sending 20 cannonballs off at once. it means it's sending 20 off in rapid succession. So the little graph here is kind of trying to illustrate that. A weak stimulus is getting 9 cannonballs from this cell in this short period of time, but a strong stimulus is getting, 20. So it's that difference in firing rate that's important. Okay, is everybody good on this question?
What do you mean by cannon accidentally goes off?
So if you think about our happy little pirate friend, he's accidentally sending a signal. Yeah, so it's loaded, ready to go, and he accidentally trips a fuse on it. We'll go through a couple examples of what it means for your perception, but it just basically means it sent a signal when there's nothing there. Good. Okay. Any questions on this? Good to move on? Okay. So now we're getting into the sensation review, and hopefully this is familiar from Monday. So this is kind of illustrating you sensing something out in the environment, it going to your perception. So in this case, it's after lunch. We could all use a siesta, so we drink coffee. And so you're making the coffee, you smell it, you hear it percolating, you see the coffee. All of these things are sensations. Let's go with looking at the coffee. When the light from the coffee hits your eye, that information is transduced into neural energy. And this neural energy then goes on to the brain for further processing, which ends up in your perception. Okay. So when it goes stimulus to transduction to perception, we call that bottom-up processing. And when prior knowledge, prior thoughts, conceptions, memories, attributions come into play and influence the lower processing, that is top-down processing. And so I say lower processing, higher order processing a lot. Basically, if you tilt your head forward, that's up and that's down. So lower processing is stuff happening in your eyes, kind of on the way back to the brain. Higher order processing is then stuff that would be towards the back of your head. Visual cortex, also frontal cortex, stuff like that. But for the most part, it works. Any questions on this? Okay. Here is the eye, also from Monday. Won't go through all the parts again, but just call your attention to the retina and the fovea. The retina, hopefully you remember, is where the photoreceptors are. Photoreceptors being the light-sensitive cells. You have rods, you have a bunch of cones, all of that happy stuff. The fovea then is the point where light gets focused at the back of the eye. This is your point of highest acuity, so sharpest detail vision. You have your best color vision there. Yeah, fovea is good stuff. Another thing that you didn't really cover is the blind spot. And the blind spot is a part where all of this information is leaving the back of your eye. So all of these photoreceptors send their little fibers down into this part where it funnels on to the brain. And because there's all of this junk crammed in there, there's no photoreceptors. Because there's no photoreceptors, you can't see. It's an actual blind spot in your eye. So this next picture is an actual picture of somebody's retina. And if you ever get a chance to look in somebody's eye, it's really cool. So the central dark spot is the person's fovea. And it looks darker than everything else because it's packed so tight full of these light-hungry cells that they're actually making it darker. The light's not getting back to the camera, and so it looks darker on the image. Likewise, the blind spot looks brighter than the rest of everything. It doesn't have any of these cells that are specially designed to pick up light information, so it's just bouncing right back off them. Does that make sense?